Good morning and good afternoon, everyone. I'm Jamie Parrott, Vice President and Chief Operating Officer for MedResults Network, and I couldn't be more pleased at the overwhelming turnout we've received for this presentation. As you're aware, we launched our partnership with Prolenium earlier this year, and the feedback that we've received from members has been absolutely exceptional, so we are truly thrilled to be working with them to provide additional education on Revanesse Versa today. Before we begin, I'd like to spend a minute or two providing a brief overview of MedResults Network, as I know we have quite a few attendees on the line today who are referred to the presentation by their Versa reps, but may not be totally familiar with what we do at MedResults and how we're aligned with Prolenium. So for those of you that are new, we want to welcome you, and for those of you that are joining us as MedResults members today, hopefully this will just be a great refresher for you. To give everyone a brief background, MedResults was formed back in 2008 with the sole purpose of providing cost savings and education to aesthetic medical practices on all the products and services they use frequently in their businesses. And we started in 2008 with 30 medical spa members. Today we have nearly 31 medical practices in the network, so we've grown significantly thanks to great partners and affiliates like Prolenium. For those of you who are familiar with group purchasing organizations or GPOs in the reimbursable space, you'll understand that we have essentially the same model, but we're the largest and one of the only GPOs focused solely on the aesthetic space. And like a traditional GPO, membership in MedResults comes at no cost to our members, so there's really no risk in belonging to the network, only a major upside, which would be savings and free education. And using the power of our 3,000 plus members, we've been able to negotiate for benefits above and beyond what our members could receive as individuals through our 50 vendor partners. And currently we have partnerships with leaders in the industry in injectables like Prolenium, skincare, medical surgical supplies, equipment, and pretty much everything you'd expect to use in your practice. And to give you a great example, one of my favorites actually, through our national partnerships with leaders like Cardinal Health and Medline, we're regularly saving our members anywhere from 20 to 30% on their medical and surgical supplies, which, as you guys would know, equates to anywhere between $10,000 and $70,000 annually or more, depending on our members' total spend. So these are just two of our partnerships, but today we're thrilled to be able to introduce our guests from Prolenium and their HA filler Versa to our entire network and guests. Now, because we have a longer event planned for today, I'd like to dive right in with the introduction of our new speaker for Prolenium. Unfortunately, Dr. Keith Burke, who was originally going to join us for the event, is under the weather, but we have an incredible speaker to take his place today, one who was actually one of the original formulators of Revanesse Versa, knows just about everything there is to know about the product. So I'd like to introduce Dr. Timothy Lee, Director of Research and Development at Prolenium. And Dr. Lee, he has a PhD in biochemical engineering. He then did a postdoctoral fellowship and eventually became a senior scientist at Sanofi, where he was involved in the optimization of biologic processes to improve product yield. Dr. Lee has also worked as a biopharmaceutical consultant for the NIH, DARPA, and the Department of Defense on government-funded projects. And he then moved to Allergan in Irvine, California, where he served as the Director of Research and Development in formulating Botox for different medical indications, such as osteoarthritis, reticular back pain, and migraines as well. And as I mentioned, Dr. Lee is currently the Director of R&D at Prolenium, and he's working on new hyaluronic acid formulations and bead technologies to bring these products to the market. He also has published several patents, as well as established peer-reviewed journals, and he presents extensively all over the world, and he's been doing that for a few years now, outlining the work performed in the industry. So we're very excited to have him here with us today. He's full of a wealth of information. And finally, as a reminder, we have so many new guests on the line today. So if you have a question for Dr. Lee, please hold your questions to the end or simply type them into the webinar chat feature, and we'll try to get to those at the close of the event. Also, we have a brief 4-question survey that automatically pops up into your browsers following the presentation. So we ask that you take 30 seconds to fill this out for us. This survey also acts as an opportunity for you to ask our presenter any additional questions that you may have about the product or the presentation. And if you do have questions for Dr. Burke, who unfortunately isn't with us today, we've been getting a lot of questions about his own techniques and his uses of Versa on the full face. So if you have those types of questions, feel free to reach out to us, and you can use the survey to do that. So once again, thank you for joining us today, and without further ado, I'd like to introduce Dr. Timothy Lee.
Well, thank you very much, Jamie. I do appreciate that very much, and thanks again for hosting this really important webinar, and also for everyone on the phone and hosting and dialing in rather. Thank you again for taking up some of your time and also, well, an hour of your time of your busy schedules to come in and to dial into the Revanesse Versa webinar. So without further ado, let me talk a little bit about the company and then dive in really into the Revanesse Versa product and some of the characteristics of the product, which will give you a little bit more of an understanding of what this dermal filler is all about. So let's go to the next slide. So Perlinium, we are the only and first manufacturing facility for HA fillers in North America. So currently right now we have sold over 1 million syringes. We were founded in 2002. My CEO and my direct report is Eric Kashbin. He's not here today, but he sends his regards. We are located in Canada, in a little bit north part of Toronto, in a part called Aurora, Ontario. We do sell internationally. For most part of the years, we've been selling within Canada and the EU. As of last year, we did have FDA approval for the Revanesse Versa, so we're very proud to know that we are selling in the US quite extensively now. And so we're pretty well global in terms of our product line, in terms of sales over many different countries. And really, it's really commercializing, and also what we do here in the Aurora side is mainly R&D. So we do the design development work, we do the manufacturing of the dermal fillers, HA dermal fillers, we do the testing of the dermal fillers, and we do also commercial sales. And so we do everything within the Aurora site, which makes it very much easier in terms of cold chain and also supply chain to be able to control these processes going from the bench all the way to commercial. So let's talk a little bit about Versa. Well, Revanesse Versa is, as we mentioned before, it's an HA gel, and HA stands for hyaluronic acid. It's not uncommon that most dermal fillers out there are HA and has a BDD cross-linked technology. So we do have a BDD cross-linker in the process of making the HA gel. We then make the gel in a clean room, and if you do know any of those terminologies, we have a very sterile environment where we make the HA gel. We mill it. So what milling is basically another fancier word of putting it, sizing it into a certain size that will be compatible for the skin, and then we dialyze it against the buffer, which is physiological pH, which is very helpful for the skin as well. And then we combine it with about 10% unmodified HA for extrudable purposes. That makes the product a lot smoother when you inject it into the skin. We filled it in 1 ml BD syringes, and then we sterilize it in an autoclave by moist heat. And basically what we have is a 1 ml syringe, which is the medical device, which is then being injected. The Revanesse Versa is unique in the sense that the products that we're making and the processes that we use are such that the HA product, the cross-link product, comes out as very uniform and spherical. And it helps because it not only helps in the smoothness of the skin, but it also, which I will explain a little bit later, helps in the biocompatibility side of things, where when there's something that is smooth, when there's something that's spherical, the skin sees it much more genuinely, and it's not something like a foreign body. And so that's one of the key features as to why the Revanesse Versa product is a smooth product, and it also has a very low in terms of the adverse reactions. We have a concentration of about 25 milligrams per mil of HA product. So we are actually right now, as my research tells me, I think we are probably the highest concentration out there in the marketplace in terms of concentration of HA. And really, I think as the right-hand side of the slide shows, is a very homogeneous type of filler with our technology that we have, and it really helps in terms of balancing the water content of the natural skin tissue. So it's very biocompatible. Next slide. Just want to show you a little bit about what it looks like. We talked a little bit about smoothness and spherical. These are the kind of particles that we're dealing with, and I think it would be a good idea just to show at this point of time the product in the middle and how it compares to the other dermal fillers on the market, like the Juvederm's Ultra, and also the gold standard that we often compare it to, which is the 1991 Restylane approved product. It's been quite a while, I guess. It's been about 25 years since it has been approved. But the overall feature that we want to show here is that the products that we have about a range of about 200 to 300 micron in size. It's the most ideal size from a scientific standpoint, because what you do is that when you inject it, you don't want it too small, because what happens is that the body sees it, it tends to engulf small particles, and then it's then removed. So this is the right size, where it's about 250 to 300 micron in size, which is ideally the best size for a dermal filler product for the purposes of not only biocompatibility, but also for the fact that it stays within the skin. All right, next slide. This gives you a little bit more of the uniformity and spherical shape particle. And I wanted to bring this one up because this is a key feature in Revanesse Versa. We often talk about the uniformity. We often talk about spherical shaped particles versus other types of fillers that may be a little bit more irregular or not so uniform. And the reason behind the importance of this topic is really the next slide, where in one of the papers published back in 2005, it has been shown and is showing over the years, and also definitely shows even in our clinical study, that the round smooth particles are more spherical. Because they're more spherical, they look like they're more biocompatible, and the reason behind that is because they represent and they look more like cellular structures. It has the smallest amount of surface area, so you don't get that interaction with other that inflammatory response. When you have particles which are irregular shape, you have a lot of surface area where potentially you have exposure of those surface areas that can cause inflammatory response, and there's a whole slew of reactions that happen because of that. That's, people have often heard about adverse reaction, looking at macrophage activation, all that kind of stuff. But the idea behind this technology is that you want something that is smooth. You want something that is spherical. You want something that has least amount of surface area for the skin, which makes it more biocompatible for the derma filler. Let's talk a little bit about the gel properties. Extrusion force, as I was saying before, that in our processes we make a cross-link HA, and we also put about 10% linear HA in there for extrusion purposes. We find that this is a very important test because, obviously, for obvious reasons, we want something that's easy to extrude. We want something that is easy, that is good for the physicians to be able to inject. Revanesse Versa, it's easy to inject. It has extrusion force about 3 pounds, which is very much in line with the dermal fillers out there. We do have, and we have seen dermal fillers having up to about 7 pounds of pressure. So, this is well below the amount that is needed for extrusion purposes. I will talk a little bit about Restylane later on, but not yet. I will reserve that for later purposes. Let's talk a little bit about cross-linking, because I think that's also another thing that is important. We talk about the shape, we talk about the size. Let's talk a little bit about cross-linking, because this is an important effect in the HA product, generally speaking. The most important part in cross-linking is that it increases the durability of the filler. And as you can see, this is basically, if you look on the right-hand side, we have a HA filler, which is a disaccharide or a two-sugar molecule, which is a long chain. And because it's a long chain, you have areas in which you can actually cross-link between chains. And that cross-linker is basically the linker that sort of like if I were to put it in an analogy of a house, what holds up the house? It's the structure, it puts the structure, the durability of that unit. And so the higher the cross-linking, the more chains you can make, the harder the filler becomes. It makes sense because basically the more you cross-link, the more, the stronger it gets. And as a result, it increases its durability. Now, one thing that is very important is that the degree of cross-linking is important to control. Because what you want is that you want something that is durable, but you don't want something that will give you an adverse reaction. And it has been known in literature that excessive cross-linking can lead to a very hard implant, which becomes a foreign body, which then becomes an unacceptable means of adverse reactions, like nodules and other things which can cause when you inject that into the skin. So the degree of cross-linking is important to have. You want enough, but you don't want it too much. And that becomes then a very critical process parameter to control. You want to promote the links between the different HA chains to keep it firm, but not too stiff. And basically, the degree of cross-linking is really, if I were to put it in the simple scientific terms, it's really the overall concentration of BDDE linkages in a chain per HA unit. So anyways, just to summarize, it's important, but you don't want it too much in order to avoid any adverse reactions. The other thing that is actually a big terminology in the industry right now is called the elasticity, or many of you have probably heard the G prime. Many people like to use this word in industry. G prime, which is the elastic modulus, it's something that often physicians and people tend to use to kind of compare between fillers. I would like to say today that although G prime is an important part, is not the only important part in comparing fillers. There's another thing that I would like to bring up to look at, which is important. But let me just talk a little bit about elastic modulus first, because it's something that we just want to make sure that we're all on the same page. So G prime is really the storage modulus. It's really how elastic the gel is. And if I were to look at it from a jello standpoint, it's really how you can recover its shape after you've twisted. So a lot of the G prime studies that we have are to look at the effect or how well the gel or the dermal filler can recover when you torque it, when you put some strain on it or deformation type of forces on it, how well it can recover back to its shape. Now, if you look at gel and if you look at it from a structural standpoint, there are many factors that can affect how elastic the filler is. The average size of the HA particle. So if I were to give an example, if you have a slab of HA or a slab of gel, it's pretty well elastic. There's nothing, you can't really, there's no flow to it. It's just a piece of very elastic type of gel. But if you cut it up into many different pieces, then you tend to lose its elasticity. So this is basically just to show that the elastic nature of the gel is affected by overall how big the particles are, how long they are, how the shape of the size, how the texture of the HA, the smoothness, how well it interacts together to make that structure, and all affects what we call the elastic nature of the HA product. We also talk about HA being viscoelastic. Now, this is important because HA is not elastic only, and it's also not viscous only. It contains two things. So we talked a little bit on the left-hand side. If you look at the picture down below with the gel, we talked about the G prime, which is the elastic nature of the product. There's also another part in the characteristics of HA, and that's what we call the viscous side, which is really the flow. And I said this before that you need both, and the hybrid properties of both the elastic nature and the flow is important for the dermal filler, because you want something that is resistant, that is able to withstand forces, but at the same time, you want something that is viscous enough that will be able to flow properly, that will be able to, fill the gaps or fill the crevices or fill the wrinkles. For an HA to be effective, it needs to have both. During injections, we tend to inject the HA filler, so because if it has the viscous nature, it's able to withstand the high shear rates right through a syringe needle, for example. But once it's implanted, it comes to exposure to all types of forces, low shearing forces, we call it intrinsic. Intrinsic is that basically those are the forces that go against the dermal filler when it's underneath the skin, like the fat, the tissue. And the extrinsic forces are those other forces that, for example, when you smile or when you induce those kind of forces on the face, like sleeping on a pillow, for example, that is what we call the intrinsic forces. And they have to basically be exposed to these kind of forces, and they have to be elastic enough that they will be able to recover to original shape so that it's able to give that plumb volumizing effect and not go away. So both natures are important in terms of viscoelastic. Let's carry on. Now, we talked about G prime, and one thing I wanted to say here is that these are the G prime numbers, just to compare Revanesse Versa versus Juvederm, and also Juvederm Plus, and also Restylane. And I also mentioned earlier that G prime is important, but at the same time, it doesn't tell the whole story. And what I mean by that is that it's okay to look at it that you have something that has a very high G prime, but how about it? How does it endure? We talked about durability before. How does it endure over the course of time? That's the reason why G prime doesn't tell the full story. It tells you how the structure is, but how does it stand over the course of time? How does it stand during the rain? How does it stand during the snow or the winds? And that's where we want to go to the other method that we tend to use in the lab, which is called the degradation test. And that's actually an important test. This is where we treat the dermal filler with hyaluronidase enzyme solution. It basically eats the structure of the HA. You mix it with the HA dermal filler, and then what we do is that we look at G prime over time to see that how that degradation rate, by looking at the degradation rate over time, we can see how strong or how weak the dermal filler is, and basically how we can actually compare our product against the other competitors. And so basically this urometric test is through viscosity, and you can look at the torque. It gives you a little bit of an idea of what it looks like as a cone and a cone shaped type of machine, which then comes down, presses on the gel structure, and basically through oscillation, we can then measure the G prime over time. And the interesting thing is that when you look at a product like Restylane, which has a very high G prime, when you look at the normalized degradation profile, when you look at the degradation profile over time, and what you have in the chart here is on the left-hand side, you have something like a G prime, and you normalize it so that you look at it over a course of time. The degradation profile shows very similarly to, I think it's been cut off, but basically the degradation profile seems to be very similar in a lot of the dermal fillers. Looking at Versa, which is the blue one, versus Juvederm, that's the orange one. The gray one is basically Restylane. So what we're trying to show here is that the degradation profile is actually a much better way to kind of understand the stability of your dermal filler, to take a better picture of what it is like when it's treated with chemical enzymatic reaction, to look at how well that degradation profile looks like over time is a better understanding of how durable your dermal filler might be under the skin. Now, Restylane has a very high G prime, there's no doubt about that. But it also does have what we call a very small, very low chemical cross-linking. So although it has a very high G prime, and that's caused by its technology, it has what we call the another type of physical type of cross-linking, which could be caused by lyophilizing and freeze drying the product so that it has a closeness of the dermal filler, and it may cause that high G prime. But when you chemically treat it with enzymatic reaction, it basically is very similar to the degradation profile as the Juvederms and also Versa. So I feel like the degradation profile is actually a very good way of looking at stability, looking at a way of comparing how well the dermal fillers compare to each other in the market and over time. So we did a study design. We just finished talking. Let me maybe just review. So we talked about chemical cross-linking as important. We don't want it to be too highly cross-linked, but at the same time, we want it to be cross-linked enough to be able to be durable. We talked about G prime being the elastic modulus to look at its resistance to strain and torque. And by the same time, we also talked about degradation as a good measure of looking at how durable and stable your dermal filler product versus time. And that's a good measure to compare with different dermal fillers. Now I want to talk a little bit about the study design, because the study design we just finished last year. It's 163 patients. Let me just go through the study design, which is the clinical study that we use for the Revanesse Versa. The qualified subjects, so those are people with wrinkle severity of about 3 or 4, which is considered to be moderate or severe on a scale. And these people had nasal label full, which is the NLF, just to give a little bit of definition around the acronyms. And they were treated with Versa on one side of the face and Restylane on the other side. So the side of the face for each product was randomly assigned. We had an investigator and also subject were both blinded in terms of the injection, and maximum of 2 ml per nasal label full. And we were looking at both wrinkle severity and also evaluating the Global Aesthetic Improvement Scale, so the GAI scale, for both the investigators, so the injector, and also the patients also evaluated it. And also we evaluated the adverse reaction recorded in a subject diary. In the right-hand side of this slide, we also use photographs to look at the WSRS to quantify the facial folds by visual assessment of the length, the depth of the fold, and also at the same time, GI was used to grade the overall improvement by comparing its appearance at the follow-up visits. So this is a 24-week period, and we do have visits 1, 2, 4, for example, 6 weeks during that period of 24 weeks. And so this is really the study. It's basically looking at an NLF. Just to reiterate, we're looking at the wrinkle severity, we're looking at the GI aesthetic improvement, and also looking at the adverse reaction and trying to understand how this compares with the gold standard, which is Restylane. So it's designed to be a non-inferiority study versus Restylane. And we set it up to reveal the safety profile for obvious reasons, because we wanted to make sure that the product is safe to use. We wanted to make sure that we can compare that accurately with Restylane, and it's defined as the endpoint of 24 weeks. This is an FDA-approved trial. So let's take a look at the data, which is actually very important. So this basically shows the mean change of the wrinkle severity from the baseline at 24 weeks. And what we wanted to look at in terms of this study is that we wanted the people who had moderate to severe wrinkle severity, the 3s and the 4s, to be able to improve by at least 1 degree of the wrinkle severity. So in this case, it has, and it shows the data to be non-inferior and also very similar to Restylane, where it had wrinkle severity improvement of at least 1 degree, a 1 scale, for example, and it shows you the plus and minus standard deviation. The other thing that we also wanted to look at was the GAI scale. So that's the Global Aesthetic Improvement Scale. And we wanted to compare it both with the investigator and also the patient from the baseline. And basically it shows you that it's very a lot of the cases with the Revanesse Versa, we had significant improvements. We can't say that it's superior, but I wanted to say here is that comparing to Restylane, on average, we had a higher percentage of people that had improvements in the much improved or very much improved categories of the GI scale. So as you can see here, the investigator GI was about 59.2% for the Revanesse Versa compared to 47% for Restylane. And likewise, the patient global aesthetic improvement scale was about 44% versus 31%. So it's very good. It's very good data, and we're very pleased with that. The other thing that we wanted to say that we were actually quite pleased with was really the percentage of swelling. So during the course of the clinical trial for 24 weeks, we had patients that kept the diaries. And part of the diary was also to record things like adverse reaction, things on like swelling. And what we found from the diaries and also from their feedback was the amount of swelling for the 163 patients was that Revanesse Versa had a percentage swelling of much less than Restylane, so 47% versus 71%. So it was very good. It kind of shows that what we were thinking of when we were making the gel previously, when we were looking at the shapes, the sizes, cross-linking, all that characteristics, does make sense from a biocompatibility and usability standpoint because it kind of helps show that in the clinical evidence of not only the wrinkle but also the percentage of swelling. One last thing I wanted to say here, and this is really the safety part of things, and now we finish this portion of the presentation, I can then open it up for questions and answers. We wanted to say that no subjects here in the study discontinued the study due to adverse reactions. So we didn't have any death, definitely not. We did not have any severe adverse reactions. But one thing that we actually found that was interesting when we did this study was that the reported emergent adverse events that were reported for Revanesse Versa was lower than that of Restylane. So the common injection site, for example, the hematomas, the swelling and the pain, where if you look at the numbers, they were lower than those of Restylane. So for example, hematoma was only 53%. It was about the same, but the swelling part, which was quite interesting, was lower than the control, which is Restylane, where you get 47% swelling versus 71%. And then where it becomes very interesting was around the pain side, where it was less, like 38% versus the 63%. So only 2 subjects reported non-injection TAE, where there's headache and arthralgia. But overall, it was a very good study where it showed not only good wrinkle improvement and improvement in the wrinkle severity, but also from a GI scale for both the injector and also as the patient was very much in the improved and also much improved categories. And also TAE was lower in some of the aspects such as the swelling and also the pain. So hopefully this gives you a little bit of an overview of the product, the characteristics, some of the features of the process, and also gives you a little bit of an idea of the clinical results that really show that Revanesse Versa is a good product and it's a good product that we can use. And I also want to thank you all for coming. And if you do have any questions, clinically or anything, you can contact your local representatives in the U.S. But if you do have any questions, there's also a info@proleniumus.com that you can actually send any comments or any questions about the product, any concerns that you may to that website. And with that, I conclude the presentation. Thank you very much.
Dr. Lee, thank you so much for joining us, and thank you again to all of our attendees who made it out today. This was a record breaker for us in terms of our webinar attendance and overall interest in the presentation. So I just, like I said, I want to thank everyone. I know Dr. Lee spoke primarily in reference to treatment of nasolabial folds, which obviously is approved. However, as I mentioned initially, Dr. Burke, he shared that although he regretted not being able to participate today, he would also be happy to answer any questions pertaining to his experience with a variety of different facial treatments using Versa. So once again, you can always reach out to me directly. You can certainly use that automated survey that's going to pop up in just a minute here to ask additional questions, or of course, you can reach out to your local rep or info@proleniumus.com. So thank you again. We hope to see you on our next MedResults webinar presentation, and I appreciate your attendance today. Have a great rest of your afternoon, and we'll see you next time.